Hello students. Today's objective for 10.4.16 as I am gone is going to be walking through the process of the Krebs cycle. The Krebs cycle is the second step of cellular respiration, and we have covered the first step in class with glycolysis. As we step into the second step, I want to review back to our worm experiment that we did with the ground and the non-ground watermelon from hours 4 to 21. So I didn't use the first 3 hours. I started in on hour 4 and stopped it at hour 21. The ground watermelon, so in the container with the ground watermelon, the slope was at an increase of 0.078 increasing of carbon dioxide. The slope of the oxygen decreased at a rate of 0.10% per hour. The whole watermelon, the slope of the carbon dioxide was increasing at an hour of 0.044%, and the slope of the oxygen was decreasing at a rate of 0.081. What these two would show you is not the ending rates, but the slope, so the rate that it is occurring. The rate that it occurred within the ground one was higher than the whole one. What this would indicate is that the carbon dioxide levels in the ground watermelon biochamber increased at a faster rate. The oxygen was used up at a faster rate, which hence a more negative slope per hour in the ground than in the whole. That slope of oxygen was a negative 0.081% in the whole, where the slope of the oxygen was 0.1%. So these two mean that there was more carbon dioxide and more oxygen being produced. Up until now, you guys with the glycolysis had nothing to do with oxygen and carbon dioxide. The Krebs cycle explains the carbon dioxide. So as we look into why would the worms be able to produce more carbon dioxide per hour, we can start to get into what's called the Krebs cycle. So today's objective is going to be finishing filling out your notes. On Monday, you moved onto your notes saying that moving into the mitochondria. And then it asks you to draw a mitochondria, and you can go get a mitochondria off of the Google Images and paste it into there. For we are talking about the Krebs cycle was the next blank. And it's a series of reactions in which pyruvate is broken down into carbon dioxide. And the steps to it are going to be using 8 NADs, 2 FADH2s and FADs, some ATP, and 6 carbon dioxides. What happens is the 3 carbon chain called pyruvates from our glycolysis will come into the mitochondria. As they come into the mitochondria, one of the carbon dioxides will be broken off by this enzyme called acetyl coenzyme A. This acetyl coenzyme A helps break the pyruvate, which is a blank in your notes, into 6 carbon dioxides. So it will break off one. As it breaks off one, that pyruvate here travels through, changing an ADP into an ATP. That means that when we broke off that bond, it changes it from a 3 carbon chain to a 2 carbon chain. This then releases a little ATP. We continue and we break off another carbon off the pyruvate. That will break and release that, and the reason I'm using a red lightning bolt is because that's energy. When we break a bond, we release energy. That energy produces some heat, which is wasted, but it keeps us warm, and it also can be bottled up and shoved into NADs. We continue that, and we produce 4 more NADs turned into NADHs. Then the last carbon is broken off of the pyruvate, and we no longer have any carbons left on our pyruvate because all 3 have been broken down into carbon dioxide. And when that happens, the energy is put into yet another energy molecule called FAD, and it's stored, the energy that's broken off is stored into what's called FADH2. This leads us to have it happen all again. Our next pyruvate comes in, and in that acetyl enzyme A, coenzyme A grabs it and helps break off the first carbon dioxide, and then we release a carbon dioxide again. And so we'll release another carbon dioxide, we'll turn in another ATP, we'll release another carbon dioxide, and then we'll make some more NADHs, and then we'll make some more FADH2s when we break off the last one. So as we get through the process of the Krebs cycle, what we are making is a whole bunch of ATP, NADH, and FADH2s. And how we do it is we break off the carbon bonds between our pyruvate carbons. This is why in our lab, carbon dioxide was being released. Our carbon dioxide was being released more because our worms were doing more cellular respiration because they had more carbon dioxide being released and caught by the sensor. When looking at this video and reviewing it, we just worked through the process of the Krebs cycle. In your notes, you should have all of the blanks filled out for the Krebs cycle. You might have to go back and review this video, but what I would like you to do is have all of those blanks filled out. So moving into the mitochondria and have a picture of the mitochondria, we move to the Krebs cycle. The Krebs cycle is a series of reactions which the pyruvate is breaking down into carbon dioxide. The steps of the Krebs cycle is that the acetyl coenzyme A, an enzyme, helps break down the pyruvate into 6 carbon dioxides, 2 ATP, 8 NADHs, and 2 FADH2s. This process of the Krebs cycle can be found on, say, the internet somewhere, and you can post a picture on your notes. So today's first objective was to identify the Krebs cycle. You should be able to identify it, and if you have any questions, make sure you star where you have questions at.